Okay, hi everyone. This is the lab demo for the Enzyme Lab. One second, I just need to get a glove here that I forgot. All right, so first, just a little bit of background information. The Enzyme Lab will be on Moodle. I realize I didn't upload it to Moodle just yet, but that Enzyme Lab will be there, and it'll be, I'll show you it right now. Okay, so that Enzyme Lab is going to be similar. Oh, it would be good if I shared my screen. So one second, let me share my screen, and then I'll pull that back off to show you the actual demo of the lab. All right, so for the Enzyme Lab, it's from the lab manual again, so there will be a PDF that you guys will have to follow, and you should be following along as I go through this demo. I will talk about the background information just using my one PowerPoint slide. That PowerPoint slide is also going to be on Moodle as well, okay? In terms of your assignment for this lab, again, it will be to make a mini lab report like we did with that last experiment. And so you're going to be filling in the data, again, in the tables here, but you're then going to be then making your own tables in Word, just like I said, you did for the chemical composition of lab, right? Answering the questions underneath each table, okay? So again, for each experimental procedure, you will have a table with your data, okay, based on what I show you, and then you'll have the answers to these questions underneath each one, okay? So the experiments that we're going to do are experimental procedure catalase activity, right? Effect of temperature on enzyme activity, okay? Again, there's a table. Also, you'll see with two of these, there is also a graph. Okay, so you want to graph your data, just write it in so you have that information while you're following along, but then you're going to make a graph in Excel, okay? So I can show you that quickly, how to make a graph in Excel, or maybe I'll just save that for our next class meeting just to kind of show you if need be. But again, you'll make a graph in Excel, you'll basically copy that image, or you'll save that image, save as a picture, and you'll insert it into your mini lab report. Okay, so you're making the tables, and you're also going to be making a couple graphs for this little mini lab report, right? And so exactly as they are here, right? But again, you're not going to take a picture and insert a hand-drawn picture. You're actually going to make that graph in Excel. Why? Because it's an important skill to know how to do, okay? All right, so effect of temperature, right? Now this is effect of concentration. Again, you're going to fill out that table. You're going to answer those questions underneath. And then the last one is the effect of pH on enzyme activity. Okay, so same thing. You will complete this table. You'll make your, in your mini lab report, your own table. You'll answer these questions, and then you will have a graph, like I said, you'll make in Excel. Okay, so there are two graphs that you will need in your mini lab report. There are this 5.5 factors that affect enzymatic activity. Don't worry about doing that table, okay? So again, the experiments that we're going to be doing and the tables you're going to need to include in your mini lab report are experimental procedure catalase activity, okay, table 5.1, the questions, 5.2, effect of temperature on enzyme activity, okay, your data in that table. There's also a graph and a couple questions that go along with this. The next one is effect of concentration on enzyme activity, okay? Table, questions underneath. Then the last one, effective pH. You're gonna put your data in a table and then answer those questions, but you will also have a graph to make of that as well. The last thing you're gonna include in your mini lab report is the answers to the questions, this laboratory review 5 questions. So you'll write them out, number them at the end of your mini lab report. So the only thing you are submitting is a Word document of your Enzyme Lab mini lab report, okay? You're not submitting the PDF, you're just using this as a guide and using it to fill out or write in notes as you listen to the lab demo. All right, so let's just go back really quickly, just a little bit of background information. Again, background information is all, sorry, my thing is boiling away there. Anything that I'm going to talk to you about is written in here in the background information of the lab, okay? So let me pull up the PowerPoint slide just to kind of go through that. I think it's just a little more organized, but like I said, actually, we're gonna have to refer to that diagram anyway, so I'll pull that back up. But there's basically just one slide, again, that's uploaded to Moodle. And again, we're talking about the Enzyme Lab, right? So everything, you know, is going to, we're basically working on one specific reaction, okay? And I'll show you a picture of that. I'll pull back up that PDF. It's in your lab there, okay? So just a little background to remind you about reactants, right? These are the molecules that will undergo a change. They then result in products, okay? Remember, when we talk about an enzymatic reaction, which is true for any biological reaction, we call those reactants substrates, okay? Remember that enzymes are specific, and we're gonna look at the specific activity of an enzyme called catalase, okay, in our lab today. Figure 5.1, if you take a look at that, is just that same basic figure that you guys saw in our lecture. Okay, so if we look here, I guess my one slide is probably not that handy because I think it really outlines what you were doing in lab. So the most important part is really just to look at this background information in the PDF. Okay, but anyway, that's there as well. But like I said, I think this is a little more helpful. So figure 5.1 is here, right? These are just showing you degradation and synthesis reactions, right? You've seen this figure before. The reaction we're going to be focusing on, as I said, is reaction involving catalase, okay? And it is a degradation reaction, okay? So again, you know, we basically, so a couple things, right? And again, this is all in here. I hate to kind of be reading from a paragraph. Catalase is the enzyme, okay? If you guys look here at this reaction down here, okay, you can see that hydrogen peroxide is the substrate. Catalase will break down hydrogen peroxide into water and oxygen, okay? And so what we're gonna be doing in this lab is we're basically gonna try to measure the amount of oxygen, right, in the form of bubbles that's produced. So if we see more bubbles, we would then indicate that that's an increase in enzyme activity, right? The enzyme is breaking down more substrate, okay? So that's really the basis of what we're measuring. We're measuring the amount of bubbles. It is not an exact scientific measurement in any means, but it's a way of visualizing this. And what I'm basically gonna do is I'm gonna kind of hold the tube up to the camera for you guys, and I'm gonna quickly just measure the bubble column height, meaning how many bubbles are there, okay? And I will tell you what that number is, and you'll be writing that down in your data tables, okay? Just a little bit more background about catalase in general. We find catalase in the peroxisome, okay? So remember that peroxisomes will break down molecules, right, inside the cell. And in the process will form hydrogen peroxide. Hydrogen peroxide is toxic to the cell. So it's necessary for the cell to get rid of that hydrogen peroxide. The way that it gets rid of it or breaks it down is utilizing catalase, okay? So the reaction that we are experimenting with in this lab is this reaction here, okay? Right? The breakdown of hydrogen peroxide by catalase, okay? It breaks it down again into water and oxygen. The bubbles that we see are the oxygen, okay? And we're gonna look at the effects of those things that I pointed out, the effects of things like temperature and pH on enzyme activity. If we see a lot of bubbles, that indicates again that enzyme activity is high or increased. If there's a decrease in the amount of bubbles, it indicates that there is lower activity, okay? So that's really what we're looking at. By the way, also in your lab, mini lab report, it's probably a good idea to have the answers to these questions. These questions will come back on the next lab exam, just so you know, right? If you look at this reaction, you should be able to tell me what is the substrate here. The substrate is peroxide, right? What's the enzyme? It's catalase. What are the products? The products are water and oxygen, okay? So that reaction for sure I will ask you about. I'll probably ask you about where is catalase found in the cell. The answer is in the peroxisome, okay, in the peroxisomes. All right, so like I said, you can read through this background information. I pretty much hit the high points here. So let's get going with the actual experiment, okay? So I'm going to stop this share so that you guys can just see the full camera. So you should be following along in that Enzyme Lab, in that PDF, and writing down the results as I go through them. Okay, this is just like the same way I did, we did it for the chemical composition lab. Okay, so we're starting on page 59, right here, this experimental procedure catalase activity. All right, one other thing too, I will be using serological pipettes to do some of the measurements of the volumes, just so you know, just because it's just another piece of lab. I'm gonna add some, one second, I gotta add some water to my, not like that. I gotta add some water to my beaker before it burns. To my boiling water beaker there. Okay, all right, that should be good. All right, sorry. Okay, so let me get rid of this share here, and then we'll get going. So as I was saying, I'm gonna be using some serological pipettes just to do some of the measurements, just because, again, I think I showed it to you guys in that first lab, but it's just another way of measuring volumes, particularly with this serological pipette. And again, I probably should have clear a little more space. With this, the actual max volume that you can measure is 10 milliliters. Okay, so I'll use it to do 1 milliliter, 4 milliliter. And this handy gadget that I have here allows me to just press this button to drop the liquid, and then this one to dispense it. So it's all automated, okay? I think when I demoed this to you in that first lab, I showed you this sort of manual way of actually doing it, but this is the automated, easier way, okay? All right, let me move my computers, I guess, so it's a little closer. I think that might be helpful. I'm actually gonna put it over here. I think that's probably better. I'll kind of show you as I go through here, okay? Maybe that's better. Yeah, okay. So I sort of have myself set up here. Let's start off, as I said, on page 59, okay? So on page 59, it tells me to add 1 milliliter of catalase into tube 1, right? And then add 4 milliliters of hydrogen peroxide. And then we're gonna measure that bubble height, okay? So for the catalase, by the way, I'm measuring it by drops, okay? So this is the catalase just because I have it in these dropper bottles. So it's about 10 drops, 1 milliliter. Okay, so there's my catalase in the tube. Now I'm going to add hydrogen peroxide. So I believe it tells me 4 milliliters, right? Okay, so I'm going to use this guy to measure 4 milliliters of hydrogen peroxide, and I'm going to add it to the tube. Oh, hopefully this works. It does. Whoa, that's what happened there. So there's my bubble height, which was a ridiculous amount. And now it just went into, wait a second. It just went into my, well, we're gonna call that, wow. I guess that catalase, I was worried that the catalase wasn't gonna work. So it must be. So let's try that again. So I'm gonna add a smaller amount, I guess. You know why? Maybe my test tubes are not big enough, but that's right. All right, so let's go a little slower here, just so you guys can see. But I'm basically gonna, all right, so here's my hydrogen peroxide, right? And when I add it, you can see how much it bubbles, right? And so when I added this it basically went the whole height of the test tube. So I'm gonna use that as my measurement, okay? So what happened to my ruler? So for our first one, for tube 1, right, in our first experiment on page 59, our bubble column height is going to be, and we're measuring in millimeters here, is going to be 10, we'll call it 11, 11 millimeters. Tube 1, right, which had catalase and hydrogen peroxide, 11 millimeters. Nope, I lie. Do they want me in millimeters? Let me just see how we want to measure this. Sorry, 1 second, guys. Let me just make sure I give you the right. Yeah, okay. So we will keep it in millimeters, but it's not 11 millimeters. It's actually 11 centimeters, okay? So convert that. Let's do some conversions here. You convert your 11 centimeters into millimeters, all right? Easy, just add a 0. Okay, so 110 millimeters or 11 centimeters, but let's keep everything in millimeters. Okay, so if I tell you something in centimeters, you're gonna have to be able to convert it. So we're gonna call it 110 millimeters, right? Sound good? That's for tube 1. All right, now tube 2 is going to be water and hydrogen peroxide, okay? So I'm gonna need to switch this. This is a mess here. We're gonna get some water. Okay, so again, you can kind of think about what's missing from these tubes, right? So we got water in this one, right? Tube 2 is water, right? And then it's gonna be hydrogen peroxide. Yikes, which was this guy here. Okay, and Hey, wait a second, I must have catalase in my, I've got all sorts of problems today. All right, I got some catalase in my thing, it's fine. I'm gonna switch to a transfer pipette, guys. 1 second, that's not gonna work out for me here. Make it a mess. Okay. All right, so instead of using that serological pipette, I'm just gonna switch to a transfer pipette because I'm making a big fat mess here. So tube 2, this is on page 59, tube 2. We're gonna put water. I put water in there, and then now I'm gonna put hydrogen peroxide, okay? This is actually easier. All right, so there you go. There's basically no bubbles, okay? Because our volume of liquid was here, there's no bubbles here. So water and hydrogen peroxide, your bubble column height is 0 millimeters. Think about why that makes sense, okay? All right, now tube 3 will have catalase, okay? So we got our catalase, our enzyme, right? So again, remember, catalase is the enzyme, hydrogen peroxide is the substrate, okay? In tube 1, we had our enzyme and we added our substrate and we saw a huge amount of bubbles, right? In tube 2, we had water, right? And we had hydrogen peroxide. So we had our substrate, right? But we didn't have an enzyme, okay? So now in tube 3, we have our enzyme, okay? And now I'm going to add sucrose, okay? All right, so water, so I lied. Catalase, our enzyme, and sucrose, 0 millimeters in terms of bubble height. Why? Catalase cannot break down sucrose, right? The substrate for catalase is hydrogen peroxide. Therefore, we don't see a reaction occurring at all. Okay, remember the amount of bubbles is an indication of the activity of the enzyme or the reaction that's occurring. If there are no bubbles, there's no reaction. Why? You need an enzyme and a substrate in order for that reaction to occur. Okay, so that concludes that first experiment, right? Now we move on. So again, that's on page 59. Okay, so we're completing that table. It asks you about the bubble column height, which we measured. I told you what that was for each one, tube 1, 2, and 3, and an explanation. I kind of talked about that, right? And then we're answering those questions underneath. Now we're going to move on to effect of temperature on enzyme activity. The bottom of page 59, but the actual experiment is on page 60. Okay, so let's see what happens. I already have those tubes set up in terms of one in the refrigerator. It's not really a refrigerator. It's in some ice. Okay, so it's in ice, right? It asks you what the temperature is. So for the one that's in the refrigerator, we're gonna say 4 degrees Celsius, okay? For this one that's in the boiling water, 100 degrees Celsius, okay? And for the incubator, we're gonna call this one, if I could read this, yeah, this one is, yeah, about 37 degrees Celsius. Okay, so think about 37 degrees Celsius is actually body temperature, okay? Equivalent to body temperature. All right, so again, you're gonna be completing this table, writing it up in your Word document, and then you're also gonna have a graph to make here. All right, so let's see what we got. So for tube 1, again, we've had them, I've already had them incubating for the 15 minutes in each one of those conditions, each different temperature. Now, I need to add the hydrogen peroxide, okay? So let's see what the bubble height is first for the refrigerator one. Okay, so that's this guy here. Let's turn this off before I burn the place down. So here's the refrigerator one. I'm now going to add that hydrogen peroxide to it, okay? And then we'll see what the bubble height is for this one. Whoa, this is some crazy peroxide. 2, or catalase. Usually when I do this, it doesn't work this well, by the way. 3 and 4. All right, so I'm going to quickly sort of measure that. And I'm gonna call that, sorry, one second, let me flip this around. So I'm gonna go with what it happened initially. So I will call that, go from here, 5 millimeters, 50 millimeters, sorry. Okay, so 50 millimeters for tube 1, which was in the refrigerator, okay? Now let's go to tube 2, which is at 37 degrees Celsius, okay? Let me now add the peroxide. Now that let me keep my transfer pipette actually in the peroxide so I don't mix them up. So let's go with this one. So I'm gonna have this ready actually here. So that's why it's helpful to have 2 people doing this, a lab partner, right? I don't have a lab partner. Anyway. 1, okay? 2, 3. All right. So for this one, it's about the same. So I would say this is probably 50. Let's call this 37 degree one. Let's call this one a slightly more. Nah, I think it's about 50. 50 millimeters, it's about the same, okay? So the bubble height was about 50 millimeters. Same as the other one, there wasn't really much difference. All right, now, if we go to, whoops, that's super hot, okay? Now we go to the boiling one, and again, let me take this off. 1 second here. Again, I don't want anything to be burning down. I'm not here. Okay. All right, so now this is the boiling one here, okay? And I'm gonna do the same thing. I'm gonna add that, I'm gonna keep it in the rack because it's hot. I'm gonna add the peroxide to it, okay? Okay. All right, so this is pretty easy. Well, maybe we'll give it a, I don't know. It's really nothing, okay? The bubble height here is nothing. I mean, you can even see the catalase is like clumped up, okay? So that was at 100 degrees Celsius, right? We had the enzyme in there. We boiled it at 100 degrees Celsius. We saw there was no bubbles in there, no bubbles. The bubble column height is 0 millimeters. Again, think about why. When we think about the effective temperature on proteins or enzymes in general, if we denature the enzyme, right, at 100 degrees for sure, it loses its function completely, okay? And as you could see, there were no bubbles. The reaction was not able to occur because we basically damaged or denatured the enzyme, all right? So that was tube 3. And again, we were on page 60, right? So this is the effective temperature, tube 3, 0 millimeters, okay? So again, we're completing that table. We're actually making a graph of this data as well, so we can see the effective temperature, right? It was at 4 degrees Celsius, right? Then 37 degrees Celsius, and then at 100 degrees Celsius, right? How did the enzyme activity change? And again, the enzyme activity is based on the bubble column height, okay? Those measurements I gave you. All right, now that brings us, let me again shut this guy off too. Okay, so that brings us to our next lab, or next experiment, right, on page 61, which is the effect of concentration on enzyme activity. All right, so again, we have tube 1, 2, 3. In tube 1, there is no enzyme, okay? Tube 2, there's 1 milliliter of enzyme, and tube 3, there's 3 milliliters of enzyme. So we're changing the volume or the amount of the enzyme that's in each tube, okay? All right, so in this case, it tells us to add the peroxide first, and then add 1 mil water for one, and then the other one, we're basically adding 1 mil of catalase, and the other one more catalase, okay? So we're gonna start with the peroxide. Okay, so we say, so 1, 2, 3, 4. So I have the peroxide in tube 1. Now I'm gonna put that peroxide in tube 2. Okay, so I'm putting the peroxide in there first. All right, tube 2, and now I'm adding the peroxide to tube 3. All right, now to tube 1, I am just going to add water, okay? So to tube 1, I'm gonna add 1 milliliter of water, and then we're gonna check to see what that bubble height is, okay? Not really any bubbles, right? So, I mean, we can just call that 0 millimeters, okay? Tube 1 was hydrogen peroxide in 1 milliliter of water, 0 millimeters in terms of the bubble column height. Okay, tube 2, we're going to add 1 milliliter of catalase to it, okay? So we'll sort of get ready to measure here. 1, 2, 3, 4, 5, 6, 9, 10. All right, so I'm gonna call it, it is 100 millimeters for the bubble column height, okay? 100 millimeters for that. And now for this one in tube 3, I'm gonna add, so 100 millimeters for tube 2, which had 1 milliliter of enzyme. Now for this tube 3, I'm gonna add more enzyme. I'm gonna add 3 mils of enzyme, okay? And we're gonna look at the bubble column height. It's probably gonna bubble over, okay? If it bubbles over, we're gonna call that 120 millimeters. Okay, so it bubbled over. All right, so it's 120 millimeters, all right, for tube 3. Okay, so that concludes that experiment for effect of concentration, right? Meaning on how much enzyme is there, okay? And so again, tube 1 had no enzyme, 0 millimeters. Tube 2 with 1 milliliter of enzyme, we said it was 100 millimeters, the bubble column height. And then tube 3 was 120 millimeters. There is no graph for this one. It's just completing the table and answering the questions. All right, finally, the last lab that does have a graph, so another graph for this one, is the effect of pH on enzyme activity. Remember that enzymes work at a particular optimal pH, right? The example I gave in the lecture PowerPoint was that, you know, a enzyme, digestive enzyme that works in the stomach, right, like pepsin, it works at a pH of about 2, okay, really acidic. a similar protein like trypsin, which works in the small intestine, works at a pH of about 7, okay? So pH can really affect the activity of an enzyme. Now, if we're gonna talk about catalase, we're gonna say that catalase's optimal pH is probably around 7.2, 7.4, okay? In terms of its pH. So we're gonna go with that. And then we're gonna look at, we would expect, right? If we're gonna make a hypothesis, we would hypothesize that the enzyme activity would be highest, or the bubble column height would be highest at the pH of 7, right? And it will be decreased at the pH of 3 and also decrease at the pH of 10. Okay, so let's see what happens. So again, tube 1 is pH 3, tube 2 is pH 7, and tube 3 is pH 11, okay? So I'll add the catalase to each tube, and then we'll basically just add the water that's been adjusted to those various different pHs to each specific tube. All right, so let me get that stuff sort of ready here. Oh, I thought I had another set of tubes. No, that's okay. I do here. I got 1, 4, and 5, so don't worry, I'll know which ones are which. Let me just rinse these out a second. Okay. All right, so let's add our catalase, and then we will add what we need to add. So 1 ml of catalase goes into each one. Ooh, I need another bottle of catalase. Okay. So here's my catalase. 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. All right, let me clean up a little bit. This is just like in the kitchen. If you're it's not good to be a messy chef or to be a messy scientist, because then it makes for messy data, right? So let me move this out of the way, because all this is doing is cluttering my space. Okay. All right, so let's grab. We will eventually have to add peroxide too. All right, I need one more transfer pipette, so I just threw mine away. All right. Okay, guys, so I have my catalase in there. Next, I need to add 2 mils of each of those different waters, okay? So I'm gonna do that. So I'll start off with the pH of 3, which goes into tube 1, okay? So 1. All right, so there's my pH 3 water. All right, now for the pH 7. All right, so for the pH 7, I'm gonna same thing. I'm gonna add 2 mils. 1 and 2. Okay, and then lastly for the pH 10, which goes into tube 3, I'm going to add another 2 mils, right, of water. Ooh, almost put it into the wrong tube. All right, so there we go. So now we have our water adjusted to those certain pHs. And now we're gonna add our hydrogen peroxide to each tube, and we're gonna see what happens with that bubble column heights. And somehow I moved my ruler, oh, it's right here. Okay, so let's see, tube 1 is our pH 3, okay? So let's add our hydrogen peroxide. Let's see what that bubble height ends up being. All right, so I'm going to call this pH 3. The bubble column height is about 10 millimeters, okay? 10 millimeters. For pH 3, tube 1, 10 millimeters. Now, if we go to, oh, I just inhaled something. If we go to tube 2, right, which is the pH 7, which we said is the optimal pH for catalase. Let's see what our bubble column height is there. Oh, I'm not getting what's going on here? I'm not really getting peroxide in here, sorry. All right, I probably had about the same. It's about 10 millimeters. Now, it could be that this catalase is in a, I mean, maybe a little bit more. Yeah, we'll call that 20. It's definitely slightly more. So if you see here how we're getting bubbles, if I shake this guy around, I'm really not getting any bubbles anymore. All right, so we're gonna call the pH 7 tube 2 to be 20 millimeters, okay? Tube 3 was 10 millimeters, tube 2 is 20 millimeters, and then we're gonna go with tube 3, which is pH of 10, and we're gonna see what we got there. So let me just try to use a different, let me pull out of here. I'm just gonna pull out of my, directly out of the bottle here. Yeah, well, probably should be repeating that. All right, so I mean, based on this, it looks like definitely the bubble height is higher in this. So I'm gonna call this bubble height's about 50 millimeters. Okay, so the pH 10 was about 50 millimeters. All right, so I mean, and it could, whatever. Your experiments aren't always what you expect, right? So tube 1, which was pH 3, we said 10 millimeters was the bubble height. Tube 2, which was pH 7, right? 20 millimeters. And then tube 3, which was a pH of 10, I said 50 millimeters, I believe. Okay, so those were our measurements there. So remember, you're gonna have to graph that data. All right, so we basically completed all of those experiments. And again, tables, you are making them in Word. You are then answering those questions underneath. Wherever there is a graph, which is the pH experiment and the temperature experiment, we need to go into Excel and make the graph in Excel, and then either just copy paste that graph into Word, or sometimes you may have to actually click on your graph and save it as a picture and then insert it into Word, just so you guys know. Those two graphs belong in there. And then remember, you're going to also answer the lab review questions. Okay, I know this is upside down. And that will be at the end of your mini lab report. Okay, so again, for your assignment, you are not handing in the PDF, you're making a mini lab report. And by the way, this is the last little mini lab report you'll have to make. For our next lab, the mitosis and meiosis lab is really just identifying cells in different stages of mitosis and meiosis. The lab after that is an electrophoresis and a DNA extraction lab. You probably won't have to do a lab report for that. There'll be some sort of assignment, but this is probably the last of our more, I guess, involved write-ups. All right, guys, thank you very much.